Dr. Joel Graff. Today we're doing a little something different. We're shooting in front of a live undergrad audience, but we're talking about primer design. So this is for making PCR primers. We're going to have a certain sequence of double-stranded DNA. We've got it labeled 5' to 3', 5' to 3'. And we have some sequence in the middle that's blue that we want to amplify, whereas the sequence in, I'll call it purple, we don't want to amplify, we just want this blue stretch. I have a couple lines here to indicate that this is actually a fairly long piece of DNA that we're going to amplify. Double-stranded DNA is held together with hydrogen bonds, and so if you heat up some DNA, those bonds between the two strands will break prior to any of the covalent bonds that are along the length of each single strand. So once you add heat, those strands separate, and then if you cool down the temperature, you can allow new DNA to stick onto the sequence. These sequences can either end up sticking back together, or if you have a reaction set up where the reaction is loaded with the primers that just outnumber the template DNA, then you will get the primers attaching to the DNA before your two strands get back together. So when you want to make PCR primers, you need to design what is called a forward primer and a reverse primer. Okay? This forward primer is going to be exactly the same sequence. So this is going to be the same as the primer, as the top strand that you are trying to amplify. So this sequence here, it's going to be the same sequence as here. Usually when you get a sequence, you're going to be given just the top strand sequence. So if you download a gene sequence from NCBI, for example, you're just going to get the top strand. So then the forward primer is easy to design. You just create, starting at the nucleotide that you want to start, that's at the end of your amplicon, you start putting in, then you start taking down that sequence to order your primer. You want, in our lab, we always are shooting for a TM of 60 degrees Celsius, and you can use a website called Oligo Analyzer. This is from IDT. It's in Coralville, Iowa, right across from Walmart I used to shop at. Anyway, IDT has the order, you put the sequence in and hit enter, analyze, and it'll tell you the melting temperature for your primer. If it's right around 60, that's good. All the primers in our lab start out, or we attempt to get as close to 60 degrees as possible. The reverse primer is shown down here, and its sequence is going to be the antisense to the sequence you probably downloaded from GenBank, which means that you need to take this sequence and get the reverse complement. Okay, so it's going to actually match the sequence that would be on this other strand, but since you probably have this sequence, so how do you do that? You type in this sequence and throw it into Oligo Analyzer, and it'll automatically spit out a reverse complement for you. So once you make these primers and go through many rounds of PCR, you will end up with a PCR product that is mainly double-stranded DNA that is the exact length of the gene that you, or of the sequence that you are trying to amplify. I'm calling this basic PCR primers because in future videos we're going to talk about adding on additional sequence to your primers so that there is then additional sequence here and additional sequence here that is customized however you want it to be. And this will be important for cloning using either restriction enzymes or recombinant DNA technology. So this, again, this is how you design primers, just for basic, only getting a certain stretch, no extra customized sequence thrown on.